Yes, sir. Yes, sir. Yes, sir.
Okay, sounds good.
Nice. Yes, sir.
So as Mr. Chairman said, the title of my presentation is Nanomaterials and Microscope Technologies to Engineer Muscle Tissues. Currently, I'm Assistant Professor at Rasaki Institute for Biomedical Innovation. Is it one of the recent established research center at California, Los Angeles, and it was named after the Professor Paul Terasaki, who was one of the pioneers in the organ transplantation. So a little bit background about myself. I got my PhD in material science from Tohoku University. After getting my PhD, I had this honor to work with Professor Ali Kademousseni. He was a full professor at Harvard Medical School and MIT at that time. And we work at the World Premier Institute, Advanced Institute for Materials Research, or WPI-AIMR. We work on the different research areas and mostly on the hydrogels and the skeletal muscle tissue engineers. After that, I moved to University of Toronto. I work as a research fellow and in the laboratory of the Professor Melissa Radisik. And at the University of Toronto, I mostly work on the elastomeric biomaterials and the cardiac tissue regeneration. And then I moved to the US and I started working at the University of California, Los Angeles as a development engineer, and then as an assistant professor at Terasaki Institute for Biomedical Innovation. So today I would like to talk about skeletal muscle tissue engineering and the role of the nano biomaterials in engineering functional, the muscle tissues. So in order to engineer any kind of the tissues, people often use the cells. They encapsulate the cells inside of the biological scaffolds, and they come up with the kind of the three-dimensional, the scaffold structures, and using the external stimuli like electrical or mechanical stimulation, they want to have kind of the functional tissues and for the implantation and other therapeutic, the applications. And specifically about the skeletal muscle tissues, people have used the carbon-based nanomaterial like a carbon nanotube, CNT, or graphene in order to enhance the mechanical property of the scaffold and to have like a better electrical conductivity. Because the currently used scaffold for the muscle tissue engineering, they are not electrically conductive and they are not mechanically robust. And this kind of the engineered muscle tissues, they can be implanted inside the patient body or, the people. For example, it can be used to treat volumetric muscle loss. And they could have like other in vitro applications. For example, they can be used as in vitro tissue models for the drug screening. And currently, many pharmaceutical companies are interested to use this kind of the human-based, engineered tissues to test different drugs on the different kind of the tissues in the body. And specifically for the muscle tissues, they can be used as bioactuators. And because they have this kind of the contractility and they can respond to the electrical stimulation or mechanical stimulation applied from the outside of the tissues. So in this presentation, I would like to introduce Fathil method to tune electrical and mechanical properties of the heat hydrogen nanomaterial, the composite scaffolds. So the proposed idea is based on using the dielectrophoresis or DEP. DEP is a microscope technology by which a particle in the medium is polarized. When the polarization of the particle is more than the surrounding media, the particle has to go to high electric field region. This process is called the positive dielectrophoresis or PDP. When the polarization of the particle is less than the surrounding media, the particle will move to the low electric field region, and this phenomenon is called negative DEP or NDEP. DEP has some unique, properties, and it has various biomedical applications. It is a rapid and accurate and scalable technique. Rapid, it means like by applying the AC electric field, you can just quickly, just move the particle around in the media. and it depends on the accuracy of the liquid region. You can have like a different precision to move the particles, and according to the size of the electric field region, you can have different scale of the moving the particle in the media. And as a scaffold material for the muscle tissue engineering, we mostly use the gelatin methacrylor or gel hydrogels. Gel hydrogel is in natural base hydrogels. It derives from the natural gelatin. Natural gelatin has some amino groups, and let me, if you can see my, sorry. Yeah, my pointer here. So natural gelatin has some amino groups. By reacting of the natural gelatin with methacrylate and hydrate at 50 degrees for 1 hour, we can have a conjugation of the, this methacrylate and hydrate to the, amino group of the natural gelatin. And the good point about this Gelma pre-polymer is that it has some double bonds, so it can be polymerized by applying the UV light in the presence of the photoinitiator. And as a photoinitiator, we use the ICONN-2021 is kind of the non-toxic, the photoinitiator. And by applying the UV light, we can polymerize the gel hydrogel, and we can use it for the cell culture and different biomedical applications. And specifically about the gel hydrogel, it has low ionic concentration and viscosity, so it is a very good candidate to be used in the DEP micropatterning technique. So in order to make a DEP chamber, first of all, we fabricated, the electrodes. We fabricated the platinum integrated array of the electrode on the glass side using the E-beam technology. And after that, we mounted the glass, one glass side on top of that, and the thickness of this, between the glass side and the PT electrode was 10 micrometer. And after that, we introduced the polyethylene glycol hydrogel to the, this kind of the chamber. We polymerized the PEG hydrogel, and we got the very thin layer of the PEG hydrogel on the PT electrodes. And the reason is that we want to have kind of, the cell laden, the hydrogel on top of the glass side, and we don't want any kind of the adhesion of the hydrogel system to the PT electrode. After that, we mounted the IT electrode indium tin oxide on top of the this coated PT electrode, and after that, we introduced the cell, encapsulated in the Gelma pre-polymer. So later on, by applying the AC electric field, we induced the DP forces, and in a short time, as you will see later, we got kind of the alignment of the the cells inside of the Gelma pre-polymer. After getting the cell alignment and the patterning of the cell, we just the polymerize the Gelma pre-polymer, and by this way we fix the position of the cells inside of the Gelma hydrogels. And the reason is that we cannot apply the AC electric field for the very long time, so it could harm the cell membrane, and it could induce the cell death. So we should make it very short, and after polymerization of the Gelma hydrogel, we can get like a highly aligned cells inside the Gelma hydrogels. And this kind of cell alignment is very important for like to mimic the tissue function and the tissue behavior. Because in some, the tissue types in the body, like the skeletal muscle, like a cardiac, we have seen this kind of cellular organization inside of the scaffold or ECM material. And here, I would like to show you. I don't know how it doesn't work, but in this video, I wanted to show you how we can pattern, the cells encapsulated in the Gelma pre-polymer using the DP forces. Actually, we tried like two different cells. One of them was mouse skeletal muscle cells, C2, C12, and another was HUVEC, human-based endothelial cells. And in both of the cell lines, we could see kind of, the patterning of the cells between the PT electrode band and after that polymerization of this, the Gelma pre-polymer. So after getting the aligned cells inside of the gel hydrogels, we started to culture these aligned cells. And as you can see, after 5 days of the culture, we got like highly aligned, the cells inside of the gel hydrogels. And as I mentioned earlier, this kind of cellular alignment is very important to have like a better functionality and the contractility of the muscle tissues. Okay, in the next step, we introduced the carbon nanotube. We use the multifunctional carbon nanotube here, functionalized with the carboxylic groups. And we noticed the Germa pre-polymer is a good dispersant of the CNT inside of the biological media. And in the B and C, you can see the TM picture of the pristine the CNT and after encapsulation inside of the Germa pre-polymer. And when we quantify this kind of the diameter of the CNT, we notice that we have like a bigger thickness of the CNTs inside of the Germa pre-polymer compared to the pristine CNTs. And it was confirmed with the AFM measurements as well. We also quantified the peptide conformation change for the different, the hydrogen system we have, gelatin, gelatin methacrylate and GenMos-CNT. And as we functionalize the gelatin with the methacrylate and heated group and with the CNT, we could see like a lower, the movement of the peptide change inside of the macromolecules. So it confirmed also this kind of functionalization of the gelatin or GenMa with the CNT nanomaterials. And later on, we check, the biocompatibility of this GenMa-CNT hydrogels. We encapsulated the human-based fibroblast inside of these hydrogel systems. As a control, we have the pristine germ hydrogels, and we added different concentration of the CNT to the hydrogels up to 0.5 milligram per ml. And we check the cell viability using the live dead assay 2 different days after 1 and 2 days of the culture. And we noticed the cell viability was high for all CNT concentration we examined. And we also checked, the proliferation of the cells inside of the GenMos-CNT hydrogels, and we compared it with the pristine GenMo hydrogels. And interestingly, when we added the CNT to the GenMo hydrogels, we got a little bit higher proliferation of the cells inside of the GenMo hydrogel compared to the pristine GenMo hydrogels. And the reason could be kind of, the anchoring site the CNT provide for the cells inside of the GenMos-CNT hydrogels. And this kind of anchoring site is good to induce in a kind of the proliferation and, the cells and contact inside of the GenMos-CNT hydrogels compared to the GenMo hydrogels. After that, we checked the structural property of the hybrid GenMos-CNT hydrogels. We took SCM pictures and we quantify, porosity of the GenMa and the GenMos-CNT hydrogels. And interestingly, we couldn't see any kind of the significant difference in the porosity of the GenMa hydrogels up to 0.5 milligram per ml of the CNTs. And interestingly, when we see the SCM pictures of the GenMos-CNT hydrogels, as you can see in the panel E, we have seen this kind of the CNT network instead of the GenMa hydrogels. And that could be a reason why we get like a higher, mechanical properties of this hydrogel system. And later on, I will show you we got like a better electrical conductivity as well. And in the next step, we try to align the CNT inside of the GenMo hydrogel systems. And in order to do that, again, we fabricated the digitated of the PT electrode and the glass slide. We mounted the glass slide on top of that, and the thickness was 50 micrometer. And after that, we introduced the CNT, encapsulated in the GenMo pre-polymer. We applied the AC electric field and non-uniform electric field. And after less than 1 minute, we got like a highly aligned CNT inside of the GenMo hydrogel. And the reason is that this kind of the AC electric field induced the kind of, the polarity for the CNTs, and they could, because of that, they just got aligned in the direction of the electric field. So after getting the aligned the CNT inside of the GenMo hydrogels, we polymerized the GenMo hydrogel, and we fixed the position of the CNT instead of the germ hydrogens. And as you can see, we quantified the length of the CNT bonders inside of the germ pre-polymer. And as I mentioned earlier, this kind of CNT alignment was happening in less than 1 minute. And after that, we quantified the mechanical properties of the germ CNT hydrogens for the different hydrogen system. We had the control was the pristine germ hydrogens, and we quantify, the Young's modulus of this germ hydrogens, and the Young's modulus was around 12.5 kilopascal. And also we quantified, the force deformation curve for this hydrogen. After adding the CNT to the germ hydrogens, the Young's modulus was increased to 20.9 kilopascal. And after aligning the CNT inside of the germ hydrogens, we got like even higher, the Young's modulus up to 23.4 kilopascal. And interestingly, when we checked, the force deformation curve for the different hydrogen system, for the germ CNT hydrogens, we got the kind of the viscoelastic behavior of the hydrogen system compared to the elastic behavior of the pristine germ hydrogens. And that is good for us because we want to use this kind of hydrogen for the, contracted tissues like skeletal muscle or cardiac tissues. And viscoelastic behavior is very important to tolerate, the tissue force inside of the germ hydrogen system. And in this experiment, we used the concentration of the CNT 0.3 milligram per ml. And also we confirm, this kind of the alignment and the formation of the CNT bonders inside of the hydrogen systems using the AFM pictures, as you can see in the D and E panels. And then the next step, we quantified and measured, the electrical properties of the germa and the germa CNT hydrogens. And the way we just quantify this electrical property was using the PT, electrode in the system. And actually, we had two parameters here. One of them was CNT concentration. We try up to 2.0 milligram per ml of the CNT. And we had the different system. Again, we had the pristine germ hydrogen and the germ random CNT and germ aligned CNTs. And as you can see from the IV curves, when we added the CNT to the system, we have like a better or higher conductivity of the, the hybrid germ CNT hydrogens. And the germ aligned CNTs showed like a higher electrical conductivity compared to the germ random CNT and the pristine germ hydrogens. And the reason is that we use the PT electrode in the system to quantify, the electrical conductivity. And as the, CNTs are aligned in the direction of electric field, we got like a higher conductivity in the germ aligned CNTs compared to the other hydrogens. And we confirmed this electrical conductivity of the hydrogen system using the impedance measurements, and we got like a lower impedance in the germ aligned CNTs compared to the other hydrogen system we had. Okay, so in the next step, we're going to use this kind of the hydrogen system to engineer skeletal muscle tissues. And the way to do that is, again, we fabricated, the interdigitated area of the PT electrode on the glass side. And on top of that, we used the kind of the photo mask. And after aligning the CNT instead of the germ hydrogens, we polymerized the germ hydrogens. And because of the photo mask, we could get the kind of, the group rich macro pattern for the germ CNT hydrogen system. And this kind of the group rich macro pattern is very important to load the cells and the hydrogen systems. And because of this topography, we can get like an alignment of the cells on the hydrogen systems. So after 2 days of the culture, we induced the muscle cell differentiation. So differentiation here means that the fusion of the muscle cells together and making the muscle myotubes.
Hardian, you have 5 more minutes left.
Oh, okay. And we check, the functionality of this kind of the muscle tissues by immune staining of the different biomarkers. We also check the gene expression related to the maturation of the tissues. And again, we apply the electrical stimulation, and we notice the upon applying the electric field, we can get the better maturation of the muscle myofiber inside of the hydrogel systems. And here they are like a quantification of the the contraction of the muscle myofiber in different hydrogel system. And let me skip this part. Actually, we got like a vertically aligned, the CNT in the hydrogel system. And the way to do that is instead of using the glass slide or photomask on the top, if you use the IT electrode, and if we apply the electric field to the bottom and top electrode, we can get this kind of the CNT alignment in the vertical direction. And this kind of the hybrid system is very important to fabricate the kind of the biosensor based the CNTs. And in the next set of the projects, we use the graphene also instead of the carbon nanotubes. And the reason is that we just came up with the kind of the simple way to have the stable dispersion of the the graphene nanosheets inside of the water. And the way to do that is starting from the graphite. Graphite has the multi-layer of the graphene sheets, and when we have a kind of the sonication of this graphite inside of the water in the presence of the bovine serum albumin, we can get like a decoration of the BSC to the graphene nanosheets, and we can get this kind of stable dispersion of the graphene inside of the water. And this kind of simple method is very important to have kind of the biocompatible, the nano system for the making the kind of the hybrid hydrogen system. And we compare, the presence of the, the high quality graphene using the different technique like a TM, SEM, and AFM, pictures. And also we did the density functional theory calculation, and we check, the interaction between the different amino acid in the the protein as we are interacting with the the graphene sheets, and we notice there is no like the covalent bond between these two, and this is good because it doesn't affect the electrical conductivity of the graphene. And at the final, let me explain a little bit about our current research is about the 3D bioprinting of the skeletal muscle tissues. We came up with the kind of the functional bioink using the alginate and the germ hydrogens, and using that, we came up with the functional, the muscle tissues. And in this work, actually, we used oxygen releasing nanoparticles based on the calcium peroxide. And we showed, okay, using this kind of, the biomaterial, we can have, the oxygen release for the, thick muscle tissues. And by this way, we can enhance, functionality of the muscle tissues. And let me summarize my talk. So we use the DEP technique to just get the cell alignment and different alignment of the CNT and the graphene inside of different hydrogel system. And we showed this kind of the, the hydrogel system has a better functionality to induce, the tissue function and behavior. And in the final step, I showed you the 3D bioprinting and novel, the technologies and the bioinks and the biomaterial to have a kind of the functional muscle tissues. And at the end, I would like to acknowledge my mentors, Professor Ali Kadam Hosseini, Professor Melissa Radisik, and all colleagues who helped a lot in doing these projects, and all funding agencies from the Tohoku University, IIMR, University of Toronto, UCLA, and Terasaki Institute for Biomedical Innovation. And thank you very much, everyone, and looking forward to have a discussion about that. Thank you.
Thank you, Dr. Hadian, for your nice presentation. Now the presentation is open for discussion. Audience are invited to ask questions. Any doubts? Dr. Hadian, I have one question. You have shown that cell encapsulated in hydrogel. Well, with respect to time, no, you said it's growing. Am I right?
Mm-hmm.
There was a slide actually. I don't remember the number,
Mm-hmm.
but
yeah, you mean this slide,
not
right?
this one. Go down. No, some more down. Yeah, I think this one. Yes, yes.
Okay. Yeah.
This one, ABC, if you see, I mean, is it not due to aggregation? Because you started with a number of cells, but finally it looks everything has got aggregated. Is it like that? You said the
Actually,
viability and growth of cells, no?
yeah, actually we check, two functions here. One of them is cell viability, and another is proliferation of the cells. And we use the two different assays to just examine this behavior for the cells. And for the cell viability, we use the live dead assay, and it's based on the calcium within, the 3D enzymes. And using this kind of live dead assay, we can stain, the live cells as a green color and the dead cells as, the red channel color. And as you can see, over time, we get like a, the more number of, the green cells compared to the, the red cells. It means that, okay, we have like a high, the cell viability here. And another assay we used to check the proliferation of the cells was using the MTS assay. And if when we check the, the live absorption and absorptions of the, the different, the cell latent hydrogen system, and we notice we get like a higher proliferation of the cells instead of the germ single hydrogens compared to the pristine germ hydrogen.
Okay, thank you.
No problem.
Your green synthesis of graphene is very interesting, and also the finally you're going for a 3D printing for muscle tissue issues. Very great, actually, nice applications for
Yeah,
whatever
thank
you
you.
have shown. Actually, yeah, very nice.
Thank you.
Actually,
Thank you very much. And let me know if you have
yeah.
other questions. So I know the time is very short. So, and if the people have more questions, they can contact me, and we can have more discussion about the stuff we are doing.
If there are no questions from the audience.